All right, so today we are going to look over gel electrophoresis, which is one of the technologies that we know we can use to identify and look at um, DNA fragments. So, how do scientists look at DNA? Well, gel electrophoresis is a technique that scientists use um, to look at the DNA. It is a technique that separates DNA fragments by size. Electrophoresis means to carry with electricity. So, as genetic analyzation becomes more precise, the average person may get the impression that modern geneticists have powerful microscopes in their lab that zoom into DNA molecules and reveal the information. Gel electrophoresis is a technique that allows scientists to detect sequence variations among specific DNA segments based on how fast these segments are carried by the electricity down the gel. So, there are two key facts that allow this technique to work. Nucleotide sequence differences in one DNA segment compared to another can result in different sizes of these segments. Longer segments of DNA will be carried through the gel more slowly than shorter ones. Speed comparison brings in mind a race. So let's think of electrophoresis using this race analogy. So, a gel matrix is similar to a slab of gelatin made by adding agarose to water and boiling it. Agarose will then form this gel matrix, and it is made of a complex carbohydrate extracted from seaweed. This matrix will have spaces that are then filled with water when we run our electrophoresis. The size depends on the concentration of agarose we add to the water. To prepare the gel, we weigh out the appropriate amount of agarose. We mix the agarose gel with an electrolyte solution or a tris borate TEA buffer. We heat and stir the solution and pour the solution into a gel mold. These are plexiglass trays that miss the two ends. We have to tape or replace the removable ends prior to pouring, or all the agarose would spill all over our table. We then add a comb to the end of the gel while it cools, causing holes that lets us load the gel. So we can load the gel with DNA samples that will run in the electrophoresis box. And the DNA samples are delivered using a micropipette. A gel loading step must be done with care to avoid mixing the samples. We have practiced this before, but we will practice this again. Electrophoresis uses electricity as the force to move these DNA molecules through the gel. The phosphates making the DNA molecule's backbone create a negative charge. So DNA will move towards the positive poles once the circuit is complete, right? Because opposites attract. Therefore, each of these electrophoresis gels established in a lane, therefore, each of establishes a lane for the DNA samples to go through. We can then dye the DNA samples, allowing us to load their samples into the well, and we can also see it move in the electrical current, helping to track the movement of the DNA. We typically use as a tracking dye ethylene bromate. So we can see here, right, the top and the side view of the cell. The DNA race occurring in the electrophoresis gel is fair, but it's an obstacle course. This is because the DNA molecules are forced to travel through the matrix of buffer-filled spaces or pores created when the agarose gel is formed. The agarose provides a three-dimensional lane of a random mix of small and large spaces that are found at any given part of the lane. If a DNA molecule cannot fit through a pore, it moves up or down to find a pore big enough to squeeze through. The molecule does not take a straight path to positive ends of the gel, but instead it wiggles through, kind of going up and down, not left and right, but up, down. Shorter DNA segments find more pores that they can wiggle through than larger DNA segments, meaning that they move faster. For this reason, shorter DNA segments move through their lane faster than the longer DNA segments. That is why the electrophoresis method separates DNAs based on their size. The gel has to run long enough to segment, for all the segments to separate. But if it is run too long, the smaller fragments will run literally off the end of the gel. The shorter segments of DNA move more quickly through the gel than the longer segments, resulting in the fragments being arranged in the order of size. A DNA marker with fragments of known length is typically run through the gel at the same times as the other sample. This is also called a DNA ladder. So, a quick overview of what we've looked at. Gel electrophoresis. Shorter strands of DNA move quickly through the gel than longer strands, resulting in fragments being arranged in order of size. The DNA marker with fragments of known length is usually ran at the same time of the sample so we can compare them.